But before we get into meiosis, I understand some of you have some questions about the exam. For women, because as we've discussed, in women, only one of the 4 products of meiosis produces the oocyte. So you can't make those same arguments for women. So we're going to start first with the grade distribution. So number of students that earned the 4 different letter grades. Hopefully everybody's feeling happier than you might have been. So good job, generally speaking. Right? Way over half the class got A's. Works for me. That means you understood the objectives, and that was awesome. Now, so I always do this for exams. I like to show you the grade distribution so you know where you are. Not that it's important really, but you know where you are in relative to the whole class. And then I also like to show you this. So this is the grade breakdown per question. So we had 6 questions along the x-axis. What percentage of the class got 100% on each of those questions? So over 90% of the class got all the points on questions 1 and 3. So regardless of who you are, all of you did really well on those questions, with rare exception. Question 5, on the other hand, the reason I do this is because clearly I didn't help you understand the material or how I expected the answers to be formatted for question 5. So it's clear to me that I fell short, as did some of you, on question 5. And so usually after an exam, I'll show you this information so that you can self-assess. You can go back and look at question 5 and ask, did I answer it correctly or not? And then we'll often spend more time on those topics in future classes, so I'll try to bring those topics back in at various points during the semester, and those questions, like question 5, are the ones that are most likely to show up again on the final. Because my goal is that you show me that you understand the topics, and so I provide that opportunity on the final. When I see something like question 5, it will probably come back. Because I want to see, did you master that content by the end of the semester? Any questions on exams at this point? So we're going to talk next about how you get feedback on the exams. So a few of you have asked already, but are there other questions aside from how you get your exams back? Yeah. A is 80% to 100%. B is 60 to 80%. C is 40 to 60%, etc. Yeah. And that's to try to adjust for, maybe I overcorrected. That's to try to adjust for the fact that my tests are not testing on a lot of factual recall, as you saw. So it's easier percent-wise to get an A because a lot of the questions are harder. I think I might have gone a little bit too far given that more than 90% of you got A's, but that's okay. You met or exceeded expectations. That was cool to see. Other questions? Okay, we're going to work on, Brandon, our grader, and I are going to work on, as we proceed through the term, how to give feedback on exams. For this first exam, you turned it in. You've got a copy of what you turned in. But it's hard for me to give you individualized feedback because of the digital workflow. So here's your motivation for how to figure out how to get yourself feedback. We'll get to this in a second. I'm going to post right now a Google Classroom assignment that's called an exam wrapper. So this is to help you reflect on the exam. So what this is an assignment that asks you to, now that you've seen information about the grade distributions and which questions most students did poorly on, this asks you to look at your exam that you turned in, to look at the exam keys. I haven't posted them yet, but I will today. There's a PDF of the exam key that'll go up on Blackboard. They're not there yet, so don't try to click around yet. There's also a YouTube video of me solving the problems like I do in class. I solve them on the tablet. I upload a video so you can see me as I work through the whole problem. Pick one question on your exam after you've looked at the key, looked at your exam, and tried to figure out where you went wrong. Pick one question that you think you were pretty certain you didn't get all the points on. Then you have an opportunity through this assignment to write me why you think that you got the question wrong. If you understand and show that you understand where you went wrong, then you can earn up to 50% of the points that you missed on that question. So this is incentive for, so for example, if it was an 8-point question and you totally got 0 points, you earned 0 points on the question, you can earn up to 4 points. So 50% of the points on that question. By showing me that you've reflected yourself, tried to figure out where you went wrong, and can explain why you didn't get the points you should have earned on the exam. So it's up to you to try to figure out rather, not guess, where you lost points on the exam. That's the best way for you to have motivation and try to figure out where you went astray. Look at the key, look at your exam answer, try to figure out where you went wrong. If you want to come talk to me in office hours, absolutely. And as we proceed through the term from exam to exam, the way I give feedback on the exams will change. So this is one approach we're using for this exam. This might change the next time we do an exam. Questions, concerns, comments? So you see your total grade, it's on Grade Center. You've got the key, and you've got the exam you turned in. So if you're really motivated to be a geneticist and learn genetics, you should be motivated to figure out where you lost points. I know, because I've got the spreadsheet. If you want to know, come see me in my office. I'll show you your points for every question. But I'd like you to do the work. So last time we were in class, we were talking about cell cycle. This is all material that's essential for understanding meiosis. Why is meiosis important? Honestly. Why is meiosis important? To you, personally. You wouldn't exist without meiosis? Okay, there we go. Right. And maybe less relevant to some of you, you wouldn't be able to have kids yourself if it weren't for meiosis. So you might not be at a stage in your life yet when you're interested in thinking about that, but right, we wouldn't be here. Most of the organisms on the planet, the sexually reproducing ones anyway, wouldn't be here if it weren't for meiosis. So last time we talked about the cell cycle and how DNA content changes during the cell cycle, and then we started talking a little bit about how mitosis and meiosis differ. So that was exemplified by this student's submission where they showed that the output of cell division is being diploid, or having 2 copies of every chromosome for mitosis. But for meiosis, we go from a cell that's got 2 copies of every chromosome to cells that have 1. They're haploid, and those are gametes. Why is it important for our gametes to be haploid? That is, why would some of our cells have special programs, right? Not all of our cells can do meiosis, which is good, because then my pinky can't reproduce with somebody. Why is it that some of our cells are programmed to do this special type of cell division where they divide twice instead of dividing once?
Because they need to be combined with the others, like parents.
So recombination is part of it. Meiosis produces genetic diversity. We're going to talk about that later in the semester as well. It's how we get new combinations of genes and traits through meiosis and recombination. Even more so than that, let's assume you have a diploid organism, a copy of, say, chromosome 1 from mom and dad. Diploid. So an interphase cell. Got 2 copies of chromosome 1, one that came from mom at birth, or at fertilization, one that came from dad. No meiosis. So that's also what a sperm looks like. And that's also what an oocyte looks like. If our gametes were diploid, which they're not, but if they were, what would happen?
Double the amount of genetic material.
So what happens when they fuse? Now what's your ploidy? Now you're tetraploid. So you've gone from a generation of organisms that have 2 copies of every chromosome to a generation of organisms that has 4 copies of every chromosome. And if there's no meiosis, what happens in the next generation? So that cell is the same as a cell in that generation that would be a sperm, and you'd get from tetraploidy to octoploidy, and then you go from octoploidy to hexadecaploidy. Who cares? So you see the problem, hopefully, that without meiosis, without having gametes that have half of the DNA content that normal cells have, you get this huge explosion from generation to generation in the amount of DNA in every cell, and eventually your cells burst or explode or somehow go awry because they can't hold that much DNA, because you don't have that many nucleotides in your nucleus to synthesize that much DNA. So we have to have meiosis. We have to have that reduction in DNA content from diploidy to haploidy so that when the two haploid cells fuse, you restore diploidy. So that's the purpose, the essential purpose of meiosis, to maintain constant chromosome numbers across generations. What else did we talk about last class? Just bullet points. What topics did we talk about? We talked about the cell cycle, DNA replication, division. What else did we talk about in terms of DNA replication? What did I quiz on at the start of class last time? Direction of synthesis, the direction that the polymerase reads DNA template, and there was a type of one experiment, a specific historical experiment, the Meselson-Stahl experiment, that proved that replication is semi-conservative. That one double helix divides using what enzyme? Helicase. And each of those parent strands serves as a template to synthesize two new daughter strands, and that's how you go from one double helix to two. The enzyme that does that is DNA polymerase, and it adds nucleotides to the 3' end of a nucleotide chain and reads the template strand from 3' to 5'. All factual information. You know you can look that up on exams if you need to, so you can memorize it or not. But that's what we talked about last time. DNA synthesis, the cell cycle. Is there anything particularly unclear last time about all of that that you've thought about since last class and have questions about? Okay. So today we're going to talk about meiosis and how meiosis causes issues when it goes wrong. So these are the important points that we'll talk about in class today. So there's something weird about meiosis in oogenesis and spermatogenesis. There's an important difference. We're going to talk about trisomy, which is why we have somebody with Down syndrome, picture up here, rocking the 3rd chromosome. Oh no, that's a different t-shirt. There's the t-shirt that says I'm just rocking an extra chromosome. Or keep calm, it's only an extra chromosome. So we're going to talk about nondisjunction. That's when meiosis goes wrong. That's how people wind up getting an extra copy of a chromosome. It's meiosis and a problem with meiosis. So we're going to wind up at the bottom there, genetic testing of oocytes. That's what we talked about, or what I suggested you think about at the very end of class last time. I showed a graph and I asked you, please try to figure out what are the axes on the graph. We're going to talk about that at the end of class. How does meiosis differ from mitosis? Why do you hate meiosis? Let's start there. It's what? There's more to memorize. So why is there more to memorize about meiosis? It's a great place to start a conversation. What do you hate about it? So why is there more to memorize in meiosis? There are more phases than in mitosis, so go ahead. There are 2 divisions. So it's like mitosis twice. You might start with a cell that has, again, I'm just going to use one color to represent one homologous pair of chromosomes. And in my usual nomenclature, I'm going to say that one of those you inherited from mom at fertilization, the other one from dad. So one chromosome pair. And actually, I'm going to draw those as the blobs we know they look like in interphase when we are diploid. Because as we discussed last class, interphase, G1 part of the cell cycle is when ploidy is established. That's how we geneticists define your ploidy. So we got two blobs of chromosome 1. What does this cell look like after S phase? Synthesis, you go from 2 blobs to 4 blobs. So I guess I have to make the nucleus bigger to fit the blobs in. So you've got 2 copies of the version of this chromosome that came from mom and 2 from dad. So we're temporarily tetraploid. If this was mitosis, then we just have to worry about 1 cell division. We go from tetraploidy back down to 2 diploid cells. The good news is I don't require, of course you already know, I don't require all the memorization. So we're not going to talk about diplotene, leptotene, zygotenine, whatever. You know, those words, right? You don't even have to memorize them. There's only one thing I want you to remember about meiosis in the 2 divisions, which is which order they occur in. That's the part that's always tricky. So what does this cell look like? This is a G1, say, just after S phase. We'll say this is G1 of the next cell cycle. Really, it's G2. Just say G2. What comes after synthesis, G2? What happens next? Meiosis 1, since we're talking about meiosis. So there's going to be 1 cell division. This is the one part that I want you to remember. What do those 4 chromosomes look like? So there's a metaphase version of that cell. Blobs turn into things we call chromosomes. They condense, and they line up like that. So we're going to assume that the cell's going to divide here where the red dotted line is. The first cell division is going to divide the number of chromosomes in half. So how do I write the M's and the D's here? That's the important part about meiosis. There's a particular order. Are the 2 M's on the left and the 2 D's on the right? Or is it like MD, MD from left to right? What happens after the first division? Is anybody sure?
The 2 M's and 2 D's line up next to each other on opposite sides of the cell by cross the cell to 2 cells.
That's right. So the way that the chromosomes, the sister chromatids at this point, this is what's called the tetrad. You may or may not remember. That's a tetrad, 4 sister chromatids lined up together. The 2 M's pair and the 2 D's pair. And then those pairs, dyads, of sister chromatids pair with each other to form the tetrad. So the first division puts the 2 versions of mom's chromosome 1 in 1 daughter cell, and the 2 versions from the other parent in the other daughter cell. So after the first division, you wind up getting 2, what's the ploidy of these cells then? Now we have 2 diploid cells. But this is meiosis, which means that division 2 comes next. And this is where things get a little bit strange. So now we have to talk about the difference between what happens in sperm and what happens in oocytes. In oocytes, let's say that's in red, one of these, it becomes a polar body, a primary polar body. Have you heard about polar bodies before? They don't become the oocyte. So female meiosis, male meiosis are totally different. Guys, when you have that 1 cell that undergoes the 2 cell divisions, you make 4 sperm. Ladies, not so much. You'd start with 1 tetraploid cell, that primary polar body does not become an oocyte. So it divides. It produces two haploid cells, each of which carry a chromosome from one or the other of the parents. It's not always the one from dad. This is just arbitrary. The other cell divides asymmetrically. So these three small cells are the secondary polar bodies. There's three of them. We know what they are because they're tiny compared to the oocyte. So, bottom line, female meiosis is asymmetrical. You start with one tetraploid cell, you wind up with one huge cell, that's the oocyte, and three little tiny ones. Those are the polar bodies. So you get one functional oocyte out of meiosis in women, you get 4 functional sperm out of meiosis in men. Questions about this? We're going to use this information in just a minute, so don't let it slip away.
I don't understand. When did the mom do the first division, did the first polar body? I can understand second polar body, but where did the first?
So that's where the first polar body is randomly, most of the time, randomly determined. So if this was a female myocyte, a cell that's about to undergo meiosis, it divides in half. Which of these two cells becomes the polar body is, for all intents and purposes, random. So half of the time it would be this cell that became the primary polar body. The other half of the time it would be that one. So that's arbitrary. So you never know which one of the two daughter cells after the first division will necessarily become the oocyte or give rise to the oocyte. So it's just like that. If I slide those red arrows just off the side of the screen, you're free to pick which of the two on the right is going to divide to produce two polar bodies, say the one on the bottom, for example. And the other one, so that one might divide to produce two polar bodies. That one might divide to produce one polar body and one oocyte. Or it might be the other way around. Don't know. It's random. This presents a problem. It has to do with, talked about at the end of last class, but we'll get there in a minute. Does anybody remember which chromosome you get Down syndrome if you're trisomic 4? 21. Yeah. Let's see. If my bionic eyes work, that's chromosome 21 on a karyogram. It is one of the smallest chromosomes that we have. So it's not actually surprising, it's not a coincidence, that trisomy for chromosome 21 is the one that we know most about. It's because that's the smallest chromosome. It has some of the fewest number of genes, which means you don't die at birth or before birth when you have an extra copy of this chromosome. Normally, if you have an extra copy of a chromosome, chromosome 1, for example, the biggest chromosome, or chromosome 2, 3, 4, etc., the imbalance of the number of genes you have on those chromosomes compared to what you're supposed to have, 2 copies of each gene, is so great that usually it kills the fetus. But when you get down to the tiniest of chromosomes, chromosomes 21, 22, and so forth, sometimes you can survive having an extra copy. Having that one extra copy of all of the genes on that tiny little chromosome is okay. It's compatible with life. Big chromosomes, not so much. But then there's trisomy 18, which is much rarer than Down syndrome. Why? Because it's a larger chromosome. So it's much rarer for you to be able to survive having a third copy of chromosome 18. So Down syndrome is relatively common because it involves trisomy of one of the smallest chromosomes. the bigger the chromosome, essentially, the less likely it is that you can survive having an extra copy.
Is that the same for monosomes as well?
So the question is it the same for monosomy? Are you more likely to survive only having one copy of every chromosome the smaller the chromosome is? That's a great question. I don't know the answer offhand, but I will definitely look into it. Of course, you and I know that you can survive monosomy 23 because we both have Y chromosomes and there's only one of those. But for the autosomes, I don't know. But I will look into it. So the goal, the functional goal here today is to figure out how do you wind up getting an extra copy of a chromosome. So that's where meiosis comes in. It's something about fertilization, or what happens right before fertilization, that contributes to an embryo having an extra chromosome. So when does trisomy occur? Does anybody happen to know the word that is involved in producing trisomies? Nondisjunction. I heard it up here in the front. So what we're talking about is nondisjunction. That is, when chromosomes don't disjoin, that is, separate properly during meiosis. So we could start with our tetrad, just looking at, again, just one chromosome. Half of those are from mom, half from dad, like usual. Okay. We got to figure out how we're going to get a gamete. How do you make a triploid individual? Somebody with trisomy. What is the end product on the right-hand side once we're done going through meiosis and figuring this out? What are the two gametes going to look like in terms of their chromosome composition to produce somebody that's got a trisomy for any chromosome? One of the two obviously has to be diploid, right? If you're going to wind up with an organism that's got 3 copies of a chromosome, one of these gametes is not haploid. One of them's got to be carrying the extra chromosome. A whole extra chromosome. It's not like 1 1/2 and 1 1/2. That's not what happens. So the goal is, how are we going to understand, how are we going to make a gamete that's supposed to be haploid but is instead diploid? What goes wrong? So here we have the start of meiosis on the left.
Chromosomes fail to separate?
Yeah, chromosomes fail to separate.
It doesn't go to the second step, second meiosis.
So what happens if you don't do meiosis 2? Okay, so let's try that. So what does this look like after the first division? If the first division goes normally? 2 chromosomes from mom in one of the daughter cells and the 2 from dad in the other. Okay. So what happens if you don't do division 2 properly? What could be improper about division 2? Let's say the bottom cell divides there. What's going to happen in the top cell that goes wrong? How do you wind up producing, after the second division, a sperm that has 2 chromosomes instead of 1? Starting with this. What's going to happen at division? So they don't separate. So where would I draw my dotted line? Here. That red dashed line. Where's the cell, relative to the 2 chromosomes, where will the cell divide? The cell's going to divide. 2 divisions, division 1, division 2. Where do you want me to draw it? You better not tell me like this. No, that's how you get half chromosomes. That's bad. Kind of like that. The 2 chromosomes don't disjoin. They're supposed to separate and go to opposite poles, but what happens if they don't separate and they both get put into one of the 2 daughter cells? What goes into the other daughter cell? What's its genotype when nondisjunction occurs? If you're a parent cell, the mother cell that's got two versions of chromosome 1, both from mom, and they both go into one of the daughter cells, what's in the other daughter cell? It's not a trick question. Nothing. 0 chromosomes. So what? 0, well, whatever. So the output of meiosis in this case would be those 4 molecules, those 4 cells. If you get one cell that's got 1 too many chromosomes, that means it's got to have taken that chromosome from some other cell. So you've got one that's diploid, you've got one that's aneuploid. It has no copies of that chromosome. And you've got 2 normal haploid gametes. Is that good? Remedial math, you start with 4 objects, you place them in 3 containers. You're not allowed to divide them, so it's got to be 1 plus 1 plus 2 plus 0. What do you think? You better have some questions for me at this point. Please. Otherwise, I've done something very badly wrong. So what happens when the diploid sperm fertilizes this oocyte that I drew up there? Right. So this combination, a normal oocyte from a mom, of course, separate individual. Mom did meiosis normally in this case. So now let's apply the information. So as I mentioned at the end of last class, people are making in vitro fertilization-based babies all the time, literally. These sorts of issues with nondisjunction are actually pretty common. They happen pretty frequently. Measurably frequently. So if we're going to do petri dish babies, if we're going to take sperm and take an oocyte and do in vitro fertilization, implant the embryo into a mom, you better make sure that we're using good quality embryos and sperm, because you probably would get a pissed off customer if they paid you to make them a healthy baby and instead they wound up getting a baby that had trisomy or aneuploidy monosomy. So it's a quality control thing, if nothing else. But what is this graph, and what does this have to do with aneuploidy, trisomy, meiosis? This is from a manuscript that wants to solve this problem. If we're going to do in vitro fertilization, let's make sure we're picking the right oocytes and the right sperm. What's on the x-axis? What does this look like? 1, 2, 3, 4, 5, so it's okay, the numbers, great. What's the last number? X. So these are chromosomes. These are human chromosomes, 1 through 22, and then X. What's on the y-axis on each of these? Each of these is a little subplot. Right, we've got 1, 2, 3, 4, 5 different graphs stacked on top of each other. But what's the y-axis in each one? It's DNA content, basically. Number of chromatids in this case. So how do all the 5 graphs differ? What are they looking at in each of the 5? So that's number of chromatids. This is chromosome number. So the whole human genome's on the x-axis. How many copies of the chromosome is on the y-axis? What are the 5 different panels labeled? What's the top one? First polar body? So what sort of a cell are we looking at? We're looking at cells from a female or a male? female, we're talking about meiosis in females. How many chromosomes is the first polar body supposed to have? 2, right? It's the first division of meiosis. You get one daughter cell that's got 2 chromosomes, the other one that's going to become the oocyte is also diploid. So this should have 2 copies of every chromosome, right? Where does it not have this? So these are actual data. The scientists measured the amount of DNA in one polar body from a female, one first polar body. Where are the problems? How many copies of 16 and 22 does this one polar body have? What else is wrong? Not enough. 1 copy of chromosome 19. And then, over there, part of chromosome 2, not the whole chromosome 2, part of chromosome 2, there's extra. So what does that look like? Maybe a duplication? Like maybe part of chromosome 2 copied and pasted itself onto chromosome 2, so now this cell has twice as much, or 1 copy more rather, 1 extra copy of that part of that chromosome. So this is scientists using genome sequencing to detect in an individual cell whether or not it's got the correct, supposedly, biologically correct number of chromosomes. So if the first polar body has these issues, where did the extra chromosomes either come from or go to? Because I guarantee you that first cell before the first division started with 4 copies of every chromosome. So where are we going to find deficiencies? So what do they do next? They look at the second polar body. It's supposed to be haploid. Is it always haploid? 1 copy of DNA across the whole genome. The polar body is supposed to be haploid. It is haploid. Thanks for the thumbs up. So if the first polar body, which is diploid, isn't always diploid, and if the second polar body is haploid like we expect, which cell has all of the other genetic issues? There's only 4 copies of each chromosome. So we've got a primary polar body, it's got 2 copies, secondary polar body, and the well, in this case, what's pronucleus? This is from the oocyte. So in this case, the scientists destroy the oocyte, which presumably, and this is an issue we're going to talk about at the very end of class. They destroyed the oocyte. They took the chromosomes out of the oocyte and wanted to know, is that where the extra chromosomes went? What do you think? We had an extra copy of chromosome 16 in the first polar body. So where is it deficient? The oocyte is lacking it. So that copy of the chromosome that's supposed to be in the oocyte is instead in one of the polar bodies. You add them all up. There's 3 in the first polar body. There's 1 in the second polar body. There is none left for the oocyte. So now you're going to make a baby that's got only, presumably if the sperm's okay, 1 copy of this chromosome, because the oocyte isn't contributing that chromosome. What about the other 2 full chromosomes that had aneuploidy? Are they going to be okay in the oocyte or not? We've got 1 copy of chromosome 19 here. We've got 2 chromosomes in the pronucleus. That's predicted. Then we've got an extra copy in the pronucleus. We've got 0 copies of chromosome 22. Would you use this oocyte for in vitro fertilization? Personal judgment call. Probably not. Because you're going to make a an embryo that's aneuploid, unless you happen to find a sperm that has exactly the opposite pattern of chromosomal deficiencies, but what's the probability of finding that? Questions or concerns? So when you sum up the first polar body, the second polar body, and the oocyte, the pronucleus, you should always total 4 chromosomes. Why is this a problem? Why don't we just do a DNA analysis? Why did they do this? Why don't they just look at the oocyte and ask whether or not it's got 1 copy of every chromosome?
Because you have to destroy the oocyte to do it.
Right. How do you get the DNA out of the oocyte to measure how many copies of the chromosome are in the oocyte? You can't. You bust open the oocyte, you take out the chromosomes, then, oh, it was the perfect oocyte to use, but we can't put it back together. That's where this paper came in. They did the simple math. You start with 4 copies of every chromosome. If you look in the primary polar body and the secondary polar body, if every chromosome sums to 3 copies in those 2 cells, what do you infer about the oocyte? It has to have the 4th version, the 1 copy, the last remaining one that's been uncounted so far. So you do process of elimination, essentially. You use the polar bodies, which are cells that don't get used anyway in fertilization. You look at how many copies of DNA are in those 2 cells, the primary and the secondary polar bodies. Do some subtraction, then you know the genotype of the oocyte. You haven't measured the genotype of the oocyte, you've inferred it by doing subtraction. So this is a big step in vitro fertilization, being able to use subtraction and our knowledge of meiosis to figure out what the genotype of an oocyte is. A good application of genetics knowledge. Everybody's okay with this? Got plenty of time. Happy to take questions. Otherwise, I'm showing you movies of worms. I probably will anyway, but...
Is there a way to detect aneuploidy in sperm, or is it just the oocytes?
Yeah, great follow-up question. So what's the problem with sperm? So sperm make 4 functional products, but there's another difference we haven't talked about between oocytes and sperm. So the thing about the polar bodies that's useful is that they stay next to the oocyte, so you know which polar bodies went with which oocyte, because they're all together in a bundle. So if you pick an oocyte, you can actually separate the polar bodies. So you know for sure that if you get 4 chromosomes in the polar bodies, the oocyte's the one, that same oocyte is the one that's missing. Sperm don't do this. So once the sperm are produced, they physically, literally go their separate ways. You never know which 4 sperm were all products of the same one meiotic event. That's why it's not as useful for doing the same approach for sperm. Okay, so I am going to show you a couple movies to motivate our next class. So this was the end of molecular genetics, well, formally the end. Molecular genetics, as you saw, was all about the molecule of DNA. Its structure, how it replicates, how it moves around in the cell, meiosis, mitosis, replication. Now we're going to do what normally genetics classes start with, which is Mendel, P's, transmission of traits. So to this point, we've been talking about the molecule of DNA. Now we're going to watch DNA move from generation to generation and how that affects individuals. So don't fall asleep yet, because I know a lot of you have seen Mendelian genetics a lot. We are definitely going into more depth and detail than you've seen in the past. So I'm going to remind you, this is the story for next class. I'm going to ask you to think about a type of experiment you need to do. I've already told you before that C. elegans can either cross, you can mate males and hermaphrodites together to make offspring, or you can take a hermaphrodite by herself, she produces her own sperm and eggs, she can produce her own offspring. So that's important knowledge to have. Now I want to show you how C. elegans normally move, because we're going to start talking about a mutation. A couple mutations, actually. So this is what normally, if you want to come over to my lab someday, I'll show you what they look like under the microscope. But this is what C. elegans look like when they move. C. briggsii, too, the species I work on. Looks kind of like a snake. It makes a sinusoidal wave, crawls around on the plate. So it finds something it's attracted to, like food, it crawls that direction. Then we have these mutants that are called roller worms. It may be hard to see, but instead of crawling on one side, they sort of rotate like a corkscrew. And so they don't sinusoidal wave motion in a straight line. They wind up going in circles, which is why you see that circle on the plate. This worm is basically crawling, rotating in circles, spinning as it rotates. It's broken. So there's a mutation in this worm that makes it not crawl like a normal C. elegans. It's called a roller mutant for obvious reasons. Okay. So here's important information about those two mutations. Roller mutants, the mutation that causes worms to corkscrew, is recessive. So the first point up there, you have to have, both copies of the worm chromosome have to have the same mutation to make a worm that corkscrews, that rolls. Okay? So that's important point number one. The second important point is about a second mutation. So you can take, and you've probably heard about this before, maybe, doesn't matter. You can take jellyfish genes, like the gene that encodes this protein we call green fluorescent protein. You can take this gene out of jellyfish and put it in C. elegans or C. briggsii, and it makes them glow green. In this case, it makes their throat glow green. Different mutations. So we've got roller mutation, we've got the GFP, green fluorescent protein mutation. These are dominant. So the presence of only a single copy of that extra gene that we've artificially introduced into worms is enough to make them green. So here's what I would love to do. I want to be able to make roller worms that also glow green, because that would be useful for many reasons. I want to make these kind of worms. I want to make worms that are rollers and glow. So if we're going to do that, yeah, we can sell them at rates. So this is what I'd like you to do. We probably won't make any money at this. But say you start with worms that have these genotypes. You've got roller worms. Because it's recessive, you know that if they are rolling, they have the phenotype of rolling in a corkscrew, that they have two mutations. They're minus over minus. And you've got a second worm that's heterozygous. It's got one copy of the GFP gene on one of its chromosomes. The other chromosome's normal, plus wild type. So think about these three questions. How can you combine the traits? Can you make a worm that has both of the traits at the same time? It rolls and it glows green like we saw in that last movie. And also that passes those same traits, that combination of rolling and green fluorescence, to its progeny and the progeny's progeny and so forth. And most importantly, what we're doing in transmission genetics is cross design. How would you take any two organisms, plants, animals, what have you, that have two traits you want, how do you most efficiently combine them together to make a corn plant that both has really large kernels and is also sweet, or a sweet worm that rolls and is green? So that's what I'd like you to do for next class. Okay? Think about it, have a great weekend, and I'll see you on Monday.